Hello friends, I am Dr. Neela Mahajan. I shall be discussing Introduction to Pulmonary Drug Delivery today. In this module, we will introduce ourselves to pulmonary drug delivery. We will discuss advantages of pulmonary drug delivery, its formulation concerns and approaches, and lastly we will be discussing aerosols for pulmonary drug delivery. Pulmonary drug delivery is one of the targeted drug delivery methods used for the local therapy of pulmonary diseases like asthma or COPD, which is another name for chronic obstructive pulmonary disease. It is particularly used to achieve systemic absorption of the administered drug substances like peptides or proteins exhibiting a relatively poor bioavailability by the oral route. Now we will discuss the advantages of pulmonary drug delivery. Pulmonary drug delivery is the needle-free method of delivering a drug which requires a fraction of oral dose with negligible side effects. Moreover, degradation of the drug by the liver is avoided and quick onset of action is ensured. Though we have learned that pulmonary drug delivery is an advantageous method of delivering drugs, yet to reap its benefits fully, it is necessary to take care of the major concerns for pulmonary drug delivery. These major concerns include in vivo drug stability, drug irritation and toxicity, transport, targeting specificity, immunogenicity of proteins, and drug retention and clearance. Human respiratory system comprises of two regions, that is conducting airways and respiratory region. Human respiratory tract is a branched system of air channels with approximately 23 bifurcations from mouth to alveoli. The major task of the lungs is gas exchange by adding oxygen to and removing carbon dioxide from blood passing the pulmonary capillary bed. The conducting airways of human respiratory system consist of nasal cavity and the associated sinuses, nasopharynx, oropharynx, larynx, trachea, bronchi and bronchioles. Respiratory region consists of respiratory bronchioles, alveolar ducts and alveolar sacs. Now discussing the pulmonary drug delivery formulation concerns and approaches. Let us discuss formulation concern number 1. The first formulation concern is the low pulmonary bioavailability of large macromolecular drugs due to enzymatic degradation or low mucosal permeability. The formulation approach for this concern is improved pulmonary bioavailability of such drugs by inclusion of various permeation enhancers such as surfactants, fatty acids and saccharides, chelating agents and enzyme inhibitors like protease inhibitors. Now discussing the formulation concern number 2. The second formulation concern is protein stability in the formulation. The formulation approach to tackle this concern is maintenance of pH by addition of buffers to dry powder formulation and reduction of chances of protein aggregation, if any, by the addition of surfactants like twine. Prevention of denaturation during prolonged storage by addition of stabilizers like sucrose to the formulation. Now discussing the formulation concern number 3. Pulmonary bioavailability largely depends on the physical properties of the delivered protein, and it is not the same for all peptide and protein drugs. The formulation approach to tackle this formulation concern is the use of the controlled release aerosols for intratracheal delivery to enhance the desired effect like insulin liposome. It also involves the modification of the physical properties of the delivered protein to prolong the pulmonary residence time by increasing the dissolution half time to enhance its absorption. Now discussing aerosols for pulmonary drug delivery. The two possible mechanisms for delivery of drugs to the lung include an aerosol or direct instillation. The most commonly used is the aerosol, which consists of finely divided liquid droplets or solid particles in a gaseous suspension. A device which generates an aerosol is termed as an atomizer. An atomizer may be electrically, pneumatically, or mechanically powered. The drugs delivered by aerosols are deposited in the airways by gravitational sedimentation, inertial impaction, and diffusion. Mostly, large drug particles are deposited by first two mechanisms in the airways, whereas the smaller particles get their way into the peripheral region of the lungs by diffusion. The devices used to produce aerosols generate a drug-containing aerosol cloud with the highest possible fraction of particles in the desired size range. The various types of devices used to produce aerosols include nebulizers, metered dose inhalers, also called as MDIs, and dry powder inhalers, called as DPIs. Nebulizers are further divided into two categories: pneumatic nebulizers and electronic or ultrasonic nebulizers. Pneumatic nebulizers are further divided into two categories: jet and hydrodynamic. Now we will discuss pharmaceutical nebulizers. These can be divided into two main groups: pneumatic and electric. An electric generator operates from an electric source, while a pneumatic generator derives its power from pressurized gas source. The properties of nebulizers vary widely, while all produce droplets with sizes in the range 1 to 10 micrometer. They vary significantly in droplet size distribution and pulmonary deposition. Consequently, most use some sort of baffle system in the airstream. Coarse droplets impact on this and are returned to the reservoir for renebulization, while smaller particles avoid the baffle and are delivered to the patient. Now we will discuss the working of the ultrasonic nebulizer. In ultrasonic nebulizer, high frequency sound waves are produced in the liquid in the nebulizing unit. The small droplets are produced by these surface waves. The droplets thus produced are conducted away by an airstream and are inhaled by the patient. Now we will discuss the working of the hydrodynamic nebulizer. Working of the hydrodynamic nebulizer involves a number of steps. The first step is preparation of a film of water for aerosol formation by flowing it over a hollow sphere, then expulsion of gas at supersonic velocity from a small orifice in the sphere, and the next step involves the rupturing of the thin film of water, and ultimately it leads to the production of a continuous dispersion of fine liquid particles. Now we will discuss the working of jet nebulizer. In a jet nebulizer, high velocity gas flow is directed over a tube that is immersed in a water reservoir. The driver gas expands and the pressure over the tube decreases. As a result, the formulation is drawn into the gas stream and nebulization is achieved due to high shear rate in the jet stream. Now we will discuss metered dose inhalers. These are also called as MDIs. In these systems, the drug is usually a polar solid dissolved or suspended in a non-polar liquefied propellant. If the preparation is a suspension, the powder is normally micronized by fluid energy milling and suspension is stabilized by the addition of a surfactant, for example, lecithin, oleic acid, and span and tween series surfactants. Metered dose inhalers are most commonly used drug delivery systems for inhalation. Propellants have a high vapor pressure of around 400 kilopascals at room temperature, but since the device is sealed, only a small fraction of the propellant exists as a gas. The canister consists of a metering valve crimped onto an aluminum can. Individual doses are measured volumetrically by a metering chamber within the valve. The valve stem is fitted into an actuator incorporating a mouthpiece. The aerosol, consisting of propellant droplets containing drug, is delivered from the actuator mouthpiece at very high velocity, probably about 30 meters per second. There is partial evaporation of propellant prior to exit from the atomizing nozzle and further breakup of droplets beyond this point caused by the violent evaporation of the propellant. This results in a wide droplet size distribution from 1 to 5 micrometers. Only 10% of the particulates delivered in a single dose released by a metered dose inhaler actually reaches the lungs, since the bulk of it impacts in the oropharynx and mouthpiece. There has been an increased interest in modifying metered dose inhalers to minimize the number of administration errors and to improve the drug delivery of aerosolized particles into the nasal passageways and respiratory airways. Some of these modifications have included the introduction of tube spacers, breath actuators, and portable plastic reservoirs with inhalation aerosols. Now let us discuss the advantages and limitations of nebulizers and MDIs. First of all, we will discuss the advantages of nebulizers. Nebulizers produce droplets with sizes in the range 1 to 10 micrometer. They may vary significantly in droplet size distribution and pulmonary deposition. Consequently, most use some sort of baffle system in the airstream. Coarse droplets impact on this and are returned to the reservoir for re-nebulization, while the smaller particles avoid the baffle and are passed to the patient. Secondly, nebulizers deliver very large quantities of drugs if necessary without major formulation problems possible by continuous nebulization, low oropharyngeal impaction due to possibility of adjusting the airflow to suit the patient's inhalation rate. Now we will discuss the advantages of metered dose inhalers. Each MDI canister can hold between 100 and 200 doses of between 20 microgram and 5 milligram of drug, which is released within the first 0.1 seconds after actuation. MDIs are available in small size and are convenient. MDIs deliver specified amount of dose. MDIs are inexpensive and quick to use. Now we will discuss limitations of nebulizers. Most nebulizers are bulky and require a fixed power source, which limits their use severely. Now we will discuss the limitations of MDIs. In case of MDIs, only 10% of the particulates delivered in a single dose released by a metered dose inhaler actually reaches the lungs, because the bulk of it impacts in the oropharynx and mouthpiece. MDIs have relatively high gas flow rate, resulting in high oropharyngeal impaction. These essentially require accurate coordination between actuation of a dose and inhalation. Lastly, it is difficult to deliver high doses using MDIs. Now we will discuss dry powder inhalers or DPIs. Dry powder inhalers are bolus drug delivery devices that contain solid drug in a dry powder mix that is fluidized when patient inhales. These are typically formulated as one-phase solid particle blends. The drug with particle sizes of less than 5 micrometers is used. Dry powder formulations either contain the active drug alone or have a carrier powder like lactose mixed with the drug to increase the flow properties of the drug. The major problem to be overcome with dry powder inhalers is to ensure that the finally micronized drug is thoroughly dispersed in the airstream. It has been recommended that patients inhale as rapidly as possible from these devices in order to provide the maximum force to disperse the powder. Quantity of drug and deposition pattern varies enormously in these devices. Modern devices use blister packs or reservoirs. So friends, in this module, we introduced ourselves to pulmonary drug delivery and its various advantages. Further, we also discussed at length the formulation concerns and approaches for pulmonary drug delivery. And lastly, aerosols for pulmonary drug delivery were also discussed. Thank you.